Good evening, ladies and gentlemen. Thank you very much. It's an honor and pleasure to introduce the first scientific speaker and the keynote of this meeting, and that is not to say that the history of science, of zoology, as Dirk Brandes gave us, is not science. Big questions approached using the smallest organisms. That is, put in a nutshell, the subject of this evening's talk, and that is also the subject of the research of Thess Bordenstein, and it's a pleasure to introduce him. I'm pretty sure he will give a very provocative talk because his credo is that microbes are overlooked and vital for what divides big organisms into species, and someone like me who is raised in the Ernst Mayerian tradition of thinking about speciation come about by geographic or formation of species by geographical factors, or if you're more trained by maybe ecological factors. I hope that will be something very new. Very briefly, I will give an introduction into his scientific career. Thess studied in the University of Rochester. To all of you who are not familiar with the 50 states in the US, that is in the northwestern United New York State, close to Lake Ontario. He did his bachelor there in 1997, his master's also there in 1999, and then his PhD study was not surprisingly, given what he's talking about today, about the end of symbiosis and speciation in 2002. He was a postdoc fellow at the Marine Biological Laboratory in Woods Hole until 2000, or from 2003, where he also became the assistant research scientist at the MBL, and then became where he is now in the Vanderbilt University in Nashville, Tennessee. In 2008, he became assistant professor. In 2013, associate professor at the Department of Biological Science. That's where he is also today. And if you look up his homepage, what I did, I found at least 17 awards and honors and grants. So that is ranging from graduate student teaching awards to the chancellors and faculty awards, and an even more impressive list of publications. I haven't counted them, you might know, maybe not, it's too long for all of us to know. It is a very impressive list, including science and tree or papers, and it's mainly focusing of the microbial-animal interaction, and that is what he's also going to talk about today. I found this very nice shortcut and ending this introduction by saying, sometimes it takes guts, or rather microbes in the guts to make a species. So take us away with your talk. Thank you.
Thank you, Matthias. That was too kind. Thank you. Hello, fellow zoologists. It is wonderful to be here. Exciting for me to hear about the science that's going to transpire over the next couple days. When you get an invitation to come to Kiel, you never say no. At least that's what I've come to learn. And one of the reasons for that is Kiel's really in the vanguard of advancing, I think, a better appreciation of the natural world, which is that zoology is not just about animals anymore. It is about the microbes inside and onside, and these are just a few of the labs that I've been inspired by when I come to Kiel, who are advancing this view along with many other labs across the world. If structure determines function, because we're so often taught that in our classes as students from cells to organs, then the animal structure is incomplete without a view of the microbial world. And so I think that is an intriguing aspect to any discipline we study in zoology, whether it be adaptation, whether it be neurobiology, whether it be biochemistry, or whether it be speciation. And so I want to begin with the luminary here, Charles Darwin. Darwin writes The Origin of Species in 1859. And a lot of evolutionary biologists like to note that Darwin's book could have been better titled The Origin of Adaptations. And the reason for that is he actually left very little to say about the origin of species or speciation. His theory of natural selection was grounded on how do you form new varieties? And ultimately, those varieties form into new species. But it was more about iterative variation arising, and that speciation was an incidental byproduct of those iterative varieties. There's one chapter on hybridization in The Origin of Species, but we don't actually have a solid foundation yet from this book. And so he actually writes, why is not all nature in confusion instead of the species being as we see them well defined? Because speciation was almost anti-evolution. That is, why would things not interbreed with each other to ultimately get a new species? And he had some actually good thoughts about why that is. I want to continue on this theme of history just a little bit to give you a flavor of how I look at the role of symbiosis in speciation. And we go as far back as to 40, 50, 60 years after Darwin. This professor, Ivan Wallin, at the University of Colorado, writes a book in 1927 called Symbionticism and the Origin of Species. And he has this quote in it, which is, "It is a rather startling proposal that bacteria, the organisms which are popularly associated with disease, may represent the fundamental causative factor in the origin of species." Now, we're not going to go that far today, for sure. But we are going to look at what is the role of bacteria in the origin of species. And for Wallin, this came from the observation, actually the first observation, that mitochondria are bacteria or bacterial derived. So well before Lynn Margulis gets credit for the endosymbiosis theory, in fact, she acknowledges Wallin in her foundational paper on this. Wallin actually makes the observation that mitochondria divide by binary fission in rabbit liver cells. And from that, he says, well, wait a minute, these mitochondria or bacteria are present in all animals and plants, and so maybe they represent some fundamental building block of evolutionary biology. A bit speculative at this point, but certainly he gets credit for making that first observation about mitochondria. So what happened to Wallin? He's not well known in history. And in part what happened is the same year, H.J. Muller does these famous radiation genetic experiments in Drosophila. And he finds that he can induce transmutability through radiation, and the variation he induces with radiation is mappable to the nuclear genome. And suddenly this begins the great modern synthesis of combining genetic variation with Darwin's theory of natural selection. 10 years later, Theodosius Dobzhansky writes one of the great books in speciation, Genetics and the Origin of Species. Dobzhansky, a fly geneticist himself, he does set the foundation for the next, nearly 100 years by giving us a biological species concept, by giving us what's known as the Dobzhansky-Muller model of hybrid incompatibilities. Dobzhansky sets the foundation. Interesting to note these two book titles. They're 10 years apart. Dobzhansky's looks a lot like Wallin's, except symbionticism is swapped out with genetics. It was almost like he was looking over there and then retermed it. So the point I want to make here is the early history of speciation biology is both eukaryocentric and nucleocentric. Eukaryocentric because Darwin formulates evolutionary biology based on observations of plants and animals. Nucleocentric because genetic variation is fundamentally linked in with these observations by Muller and Dobzhansky in the modern synthesis. And in fact, it was so extreme that even those things in the cytoplasm, the mitochondria, some were saying in a word the cytoplasm can be ignored genetically, which of course we know is not true today. So march forward several decades and we come back to Dobzhansky in this history of speciation and symbiosis because it was the sole female graduate student on Dobzhansky's team. So Dobzhansky here, his only female graduate student, Lee Ehrman. And Lee Ehrman publishes the first really good case of bacteria being linked to hybrid sterility between Drosophila subspecies, Drosophila polystorum. And what she does is she antibiotically cures hybrid sterility and was able to show that she could restore sterility of those hybrids. And she was also able to observe a proliferation of bacteria inside the testes that corresponded with the hybrid sterility. Not much happened after that, surprisingly. And it really took some advances in technology to move us to where I think we are today. And the point I'm making here is that there were big figures like Lynn Margulis who gave us this foundation of the endosymbiosis theory. Carl Woese gave us a window into the great depth of bacterial diversity, a new domain of life, and his descendants, his scholars, went off to show all the new phyla that would emerge from the technology that was created. But the history of microbiology up until this point was largely divorced from the history of botany and zoology. And then today, of course, we have sequencing technologies and a bit of an inflection point for the vast diversity of microbes that associate with animals. Okay, so what do we do? The motivational questions for our work are the following. Can host-associated microbes assist speciation? And how do we test this? What's going to be the foundation for testing this across a broad swath of both animals and plants? Very likely is going to involve removal of microbes either by antibiotics or germ-free rearing and comparing the barriers to interbreeding or reproductive isolation in the presence of microbes, a conventional state, and in the absence of microbes in a germ-free rearing or antibiotic state. What types of reproductive isolation can microbes contribute to? So across the gamut here, can they affect not only hybrid sterility, as Lee Ehrman showed, but can they affect behavioral isolation, sexual isolation, ecological adaptations? Do microbes accelerate or slow down host speciation? What's the relevance here? It's often thought because microbes are horizontally acquired that they may actually speed up the speciation process because they can come in rather quickly and change the biology. Do microbes even evolve early enough in the speciation process of their host animals or plants to play a causal role in the speciation, or do they just come in later after the speciation event has happened, and so any reproductive isolation we link to microbes is incidental, it's not causal to the speciation event? And how important or widespread is microbe-assisted speciation? Is this going to be restricted to a few rare cases that nobody cares about? Or is it potentially as common as gene-driven speciation? So here's a model for how we think about a lot of this can happen at once. So clearly we're starting with a last common ancestor here. This host population has both a nuclear genome and an associated microbiome. This microbiome is not necessarily fully tethered to the genome, but it's clearly there as part of the structural animal. And then over time, we have divergence events that lead to reproductive isolation between these two theoretical populations now. And the divergence can occur both in the genome as well as in the types or abundance of microbes that each population has. And at this point, speciation is complete when these no longer can interbreed. So microbes may come in from the environment. They may be acquired anew every generation. There may be differences between generations, and both populations will experience these kinds of dynamics. There could also be a subset of the microbes that get horizontally or vertically transferred from one generation to the next, and we do not really know the relative importance of vertical transmission of the microbiome to this horizontal transmission. We're really just scratching the surface at this problem. Okay, so once this is set up intellectually now, you can move to two types of microbe-assisted speciation. One I'm going to call the narrow sense, the other is the broad sense, okay? So the narrow sense is really just focused on microbe-microbe effects. In other words, changes in the microbiome alone can cause some kind of interbreeding problem or reproductive isolation, so narrow sense. Broad sense, as you might expect, is just a leap out to the genome now, so it takes both divergence in the genome and the microbiome to ultimately get a reproductive isolation trait. So you can imagine the relative importance of these. You may have predictions about which one being more important or not. We really don't know right now. But we've been trying to tackle these types of questions in this very large jewel wasp called Nasonia. In fact, Nasonia is pretty tiny. It's not this big, and it's not this colorful, although it does have a rather emerald sheen to it that I'll show it to you in a minute. Nasonia is one of the best genetic models next to Drosophila in insects, although not many people know about it, so I'll introduce you to it right now. So this is a video of these tiny parasitic wasps, essentially golden legs and a green metallic color on their body. A female will be subsequently chased by a male, and you'll observe some copulation, you'll observe some mating behavior here while I talk about the biology. All right, so there are 3 species of Nasonia that I'm going to talk about today. Nasonia vitripennis, or V, diverged about 1 million years ago from these 2 sibling species, Nasonia longicornis and Nasonia giralti. They diverged about 400,000 years ago. So older species pair would be vitripennis to either of these 2. Younger species pair would be these 2 here. Their distribution is both allopatric and sympatric, so you'll note longicornis and giralti are allopatric to each other, but they're sympatrically embedded within the broader range of vitripennis. It's now a genetic model. We have RNAi technology, we have full genome sequences, we have thousands of molecular markers to do mapping, we have germ-free rearing. It's really a great system. Okay, so this is the X-rated portion of the video. This is a male obviously copulating with a female. The male has to actually do a post-copulatory display in order to complete the fertilization event. If he does not come back and say goodbye, essentially, that female will not use his sperm. Okay? He has to leave some roses. Okay, so the first microbe story that I want to talk to you about is Wolbachia. And Wolbachia has gotten a lot of attention recently for its role in vector control programs across the world now, with the Eliminate Dengue project being one of the most prominent. Wolbachia are being put into mosquitoes. The mosquitoes with Wolbachia can't actually tolerate, or they don't allow dengue virus and Zika virus to replicate that well. These mosquitoes get released into natural populations and effectively replace the ones that can transmit viruses with these Wolbachia-infected ones that can't. All right. Wolbachia in Nasonia and many other insects polarizes towards the posterior end of the embryo. This is a maternally transmitted symbiont that comes from the mother's ovaries directly into the developing oocyte and then co-develops with the egg. So in blue here is the mitotically stained Nasonia genome dividing, and in green here are the Wolbachia. They also occur in the testes, so they're primarily reproductive tissue infections, and you'll see the red Wolbachia populating the testes, seminal vesicles of the male. And if you take a cross-section through the testes, and you look at a thin electron micrograph, you look into the sperm, actually these pinwheel structures are sperm tails, just a slice through them. And if you zoom in a little further, you can see Wolbachia cells populating the testes tissues. And so this is a Wolbachia cell. It's about 1 micron in size. They coexist with the sperm. They also modify the sperm, as I'll talk to you about in a second. And then inside the Wolbachia is a very large bacteriophage or viral population. This Wolbachia actually has about 60 particles in it. It surprises me that this cell hasn't lysed open yet, but eventually it will lyse. We do see active lysis happening with the phages busting open the Wolbachia cells. If you zoom in, this is what these phage particles look like, a pretty standard phage particle for all intensive purposes. Okay, the first indication that something interesting was happening with Wolbachia and Nasonia actually dates back to 1968, post-fertilization effects of incompatibility factors in Mormonella, which was the previous genus name for Nasonia. And what they were describing, and what we can still describe today, is this phenomenon of cytoplasmic incompatibility. And it's a way that Wolbachia modifies reproduction to spread itself. And let me tell you why. So this is an infected male with Wolbachia crossed to an uninfected female. Now remember, Wolbachia are in the testes, so they're modifying the sperm, fertilization occurs, and there's no or very few offspring that are produced in this particular cross. The reciprocal crosses go quite fine, and the self crosses are also fine as well. Remember that Wolbachia is also in the embryo, and something happens in this cross where the modification of the sperm is rescued in the fertilized embryo, and so this cross produces quite fine. So why would Wolbachia cause this? Essentially, it's a suicide phenotype where males are used to sterilize the females, particularly uninfected females who do not transmit Wolbachia. Only the infected females do, because this is a maternally inherited bacteria. So this can rapidly spread the infection because it imparts a relative advantage to the infected females who mate with either male, but the uninfected females can only mate in this particular cross. So 50% infection frequency in the parents, in the offspring, a 66% infection frequency. This ratchets itself every generation to ultimately go to fixation. This is what's happening in the fertilized egg here. So cytologically, the sperm genome and the egg genome come together. During pearl metaphase, we start to see chromatin condensation differences. This is the paternal chromatin, this is the maternal chromatin, again here. This is the well-known cytological defect, that the paternal chromatin doesn't condense to go through the first mitosis. The maternal chromatin does. There's a miscommunication here. Some modification from Wolbachia has brought this in. And ultimately, during telophase, there's this chromatin bridging happening, which typically leads to a lethal embryo. So that's why these eggs or the offspring are reduced here. Okay. As an extension of this unidirectional one-way crossing incompatibility, there's also bidirectional incompatibility. Essentially, if you have genetic divergence in Wolbachia strains, infected male 1 crossed to infected female 2 produces incompatibility, and it happens in both directions. So what's happening here? There's a modification effect and a rescue effect, and if there's differences in those components, those two individuals or populations, if they have different Wolbachia infections, no longer interbreed. And this is where speciation becomes relevant, because Wolbachia could potentially drive a reproductive isolation event, a speciation event, all on its own. All right, and that's exactly what we observed in the Nasonia complex. In fact, all the Nasonia species suffer from Wolbachia-induced incompatibility. So this is the older species pair, younger species pair. The interspecific parental crosses are shown in the middle, and their offspring output, there's none here and very little here. Just showing you the Wolbachia-infected testes again to remind you that this is a cytoplasmic incompatibility-induced problem between the species. And if you cure the Wolbachia infections, leave the rest of the microbiota behind, you can restore compatibility significantly. And in fact, you can actually restore compatibility even more than the parental crosses, indicative of some kind of hybrid vigor here between these younger species pairs. So this is a narrow sense model of speciation. Microbe-induced incompatibility can seal off gene flow between the animal species, all by themselves. No host genetics are needed here. But is it relevant? Did it happen early in the speciation event? And so to sum up a lot of work in one slide, I just want to show you that we've looked at a multitude of reproductive isolation traits, if they exist or if they do not exist. And so the older species pair has a plethora of isolation barriers. If you take one away, the rest are going to be there. It's really difficult for us to say which one evolved first. So all of them have equal importance, but also all of them don't have much relevance because we don't know which one came first. In the younger species pair, we're close. There's actually very little premating isolation. The majority of the sealing off of gene flow comes from the Wolbachia-induced CI. So in this case, within 400,000 years, different Wolbachia infections sealed off gene flow between these young species. Now, if you get rid of the Wolbachia, what do you call those things? Are they species? Well, now they can interbreed. So this is an infectious form of reproductive isolation, and perhaps one of the best cases that exists today. So just to put this on a schematic, microbes can evolve early in the speciation process. You can imagine a correlation here between genetic divergence over time of two species and the total amount of reproductive isolation. 0 is no reproductive isolation, complete breeding. 1 is complete reproductive isolation, species status. So the older species pair moves over here because there's enough genetic divergence that there's multiple isolation barriers. It's an old species pair. The younger complex comes down here. It's somewhere in the process of speciating. And what Wolbachia has essentially done is come in early in the process and pushed the isolation almost to completeness. So it advanced the speciation process faster than, let's say, the gradual evolution of genetic mutations accruing that lead to other reproductive isolation barriers. Fine. Is Nasonia rare? Not necessarily. There are other cases that are coming on board. So there are studies by John Janicki in mushroom-feeding Drosophila. where they've gone out into populations that are sympatric in a small zone, a hybrid zone, and they've been able to show that cytoplasmic incompatibility is happening in the direction of Drosophila recens males crossed to subquinaria females. But it doesn't happen in the reverse. So these are uninfected populations, infected populations. But there's a whole bunch of other isolation barriers that compound what Wolbachia is doing to ultimately reduce gene flow pretty significantly. So this is a nice study because it shows that it can take multiple isolation barriers all in the young time window to get a significant sealing off of gene flow. It's also in the wild and natural populations. Okay, so this is Wolbachia. Wolbachia, there are other cases coming on. There's a few other cases. But one of the criticisms of this particular area is Wolbachia is just one symbiont. It lives in arthropods. It infects 40% of all arthropod species. That's a pretty significant number. But it's not all animals. So we were thinking about how to generalize speciation and symbiosis. We simply went to something that all animals have. They all have a microbial community, and most of them have some form of a gut microbial community. And that's where we wanted to go next in terms of broadening out the idea that symbionts can assist reproductive isolation. So in terms of the gut microbiota, one question that we were initially thinking of is what guides the assembly of gut microbes across host taxa or closely related species. And you can run the gamut here. There's diet effects, there's genetic effects, there's a lot going on. This diet study showed that you could take one strain of Drosophila, the same strain, put it on different media. Those flies now acquire different microbes, and when you try and mate them back together, there's significant pre-mating isolation within a generation, and it persists for multiple generations. This was work by Sharon et al. In 2010, which essentially showed a dramatic level of reproductive isolation just through the gut microbial community. On the other hand, host genetics affects the assembly of the microbiota. So if you take a fish microbiome and you put it in a mouse, the mouse will retool that fish microbiome to look something more like the original mouse microbiome. So there's a host by microbe genetic or biochemical communication that's happening to restructure the microbes. And all these things are happening at once. And it's relevant to human biology as well. So this is a study by Bleckman last year, which showed a correlation between host genetics, human genetics, and stool microbial community changes. This is just beta diversity and ecological metric of community differences in their microbiome. There's a lot of variation here, but there's still a very significant correlation between the two. And you could sort of jump to the conclusion that genetics affects the microbiota, but of course humans aren't well-controlled model systems, so there could be an environmental effect here as well, cultural differences, geographic differences, food differences. So when natural population studies are done, or human studies are done, we often end up with this question of, is this relationship driven by the host, or is it driven by the environment, or a combination of both? And this appeared to be a relevant challenge to thinking about speciation in the microbial community. So one of the first things we wanted to do is get rid of the variation. We wanted to bring in a system like Nasonia, a laboratory model, and control for as much as possible to look into and excavate the host effect on the microbiota away from the environment. So we can do that in really careful experiments where we're controlling the diet, the gender, the age, and the only experimental variable is the host genetic background. And we're doing this with multiple species, essentially rearing them in a common environment to rule out any potential environmental variation. Now, you could expect two things in this case about how the microbiota assembles in a controlled case. It could be neutral. Hubble's neutral theory applies here to some degree, which is the assembly of the microbiome has no functional consequence. And so it accumulates randomly with respect to the nuclear genetic differences. No pattern here. The alternative would be that there is a host filtering mechanism where host microbe interactions structure the microbiome, and it may be in a predictable way, such as that the genetic divergence or similarity covaries with the microbiota similarity. And if that's the case, then we can lean into this observation as evidence for host by microbiota interactions. And if this occurs, we should be able to measure it through a tree-like comparison. I say tree-like because what we're doing here is we're looking at the evolutionary genetic differences in a nuclear marker or mitochondrial marker and asking, does that covary with the microbial community differences? This is a community metric against a genetic metric, right? So what's represented here is a dendrogram, or a hierarchical clustering relationship, of the microbial communities from two species, and we're asking, do they covary with the host genetic relationships? This is not coevolution, this is not co-speciation, it's not co-divergence. This is something new, right, where ecological communities are structured relative to the host genetic differences, a phylogenetic signal in the microbiome, or phylosymbiosis. Okay, so in Nasonia, at least in the pupal stage, if we stain for bacteria, we see a massive collection of gut bacteria in the hindgut. A lot of waste processing is happening here, and it may be a rich environment for microbes to hang out. The gut bacteria in Nasonia are dominated by this green taxa, a lot of abundance of these particular microbes, which is a gamma proteobacteria. And in Drosophila and in bees and other insects, gamma proteobacteria is a very common phylum of bacteria that lives inside insect guts. This is in contrast to mammals, humans and mice, where we see a lot of Firmicutes and Bacteroidetes. Okay? If we zoom into the diversity within that gamma proteobacteria phylum and ask, well, what does the species or OTU level diversity look like? And we're doing this across development now. What we see is, in these pie charts, the relative abundance of each species, or OTU, and n equals the number of species, or OTUs, we see. And this diversity dramatically increases in almost a microbial succession over development. So increasing diversity over increasing developmental stages. And if we put this all together in a dendrogram analysis, the dendrogram analysis shows us developmentally staged specific patterns, right? So the pupal community is different from a community relationship perspective to the adult microbial community. However, within each of those developmental stages, we see evidence for phylosymbiosis. Namely, the Giralti and Longicornis younger species pair shares more similar microbes than they do to Vitropenis, in both the adults and the pupa. So a reminder, this is the nuclear phylogeny of these particular species. We could not measure this with larvae in this case because we only had 3 OTUs. It wasn't a statistically robust analysis. They don't show any pattern, but when we sequence deeper with the larvae, with high throughput sequencing, we can recover this phylosymbiotic pattern again. Okay, so is this relevant that we see this pattern? It's really just a pattern. It doesn't indicate process yet, nothing about evolutionary biology. So is it consequential or not? And one of the ways we're thinking about this is we now have these species, and we can move the genome and the microbiome together and ask, is there a functional consequence? Not through hybridization, but through transplants, where we're moving microbial communities from one species into a germ-free state of another species, and vice versa. Right? So the prediction might be that, well, if there's some functional relevance here, it's not just all neutral, that there'll be a mismatch reduction. These organisms will suffer in fitness in some way, performance or fitness. So here's some data on that. This is development. We're measuring the rate of larval pupation, or larva to pupa. And this is, in fact, real-time video of that process. And we're just asking, when a host receives an interspecific microbiota versus an intraspecific microbiota, are there differences in the pupation rate? And there are, and they're consistent with a genome by microbiome interaction. That is, the resident microbiome does better in its resident genetic background than a foreign microbiome does. Okay? So development is slowed. Doesn't say anything about fitness yet, so we also looked at survival. And what we're looking at here is the recipient Nasonia species, either germ-free and white, it receives its own red Giralti microbiota, it receives a Vitropenis microbiota or a Longicornis microbiota. Again, these are germ-free. And in each case, for both Giralti recipients and Vitropenis recipients, receiving an interspecific microbiota reduces survival, larva to adult, in a significant fashion, from about 25 to 45% reductions in adult survival. So based on this kind of data, it's sort of like moving mitochondria between different host species backgrounds and asking, is there adaptation there? This suggests that phylosymbiosis is at least beneficial. It is at least uniquely tuned to its species background under these laboratory-controlled conditions. All right? So a microbiome on a whole could potentially be viewed as beneficial in quotes. There may be competition within the microbiome. Microbes could be competing with each other, killing each other, but the microbiome as a community could, in quotes, be beneficial to the host background. All right. So we've also done a lot of work with larvae and looking at these interactions in the hybridization event between younger and older species pairs. So this is the microbial community dendrogram of the larva microbiota. This is the nuclear phylogeny of the wasp against a distant outgroup flies here. And what I want to point out is Longicornis and Giralti, the more closely related species, share more similar microbiota. Vitropenis and Giralti, the more divergent species, also show some similarities but also some differences. When we make a hybrid between Vitropenis and Giralti, we see 90% lethality in these particular hybrids. And just before the hybrids die, we measure their microbiota, and the microbial community is significantly altered. It looks more like a Longicornis microbiota than it does a Vitropenis or Giralti microbiota. So you could reason that perhaps there's some causal association here, that the microbiota breaks down and that this leads to some kind of hybrid lethality. So we're looking at F2 hybrids. F2 hybrids are a unique advantage of Nasonia. Parents are mated. We get F1 hybrid females. In a haplodiploid organism like Nasonia, the F2 hybrids that result from these virgin F1 females are recombinants. They're haploid. They express all recessive incompatibility factors. A really good advantage for studying hybrid incompatibilities, which tend to be recessive. And in this state, we see this 90% hybrid lethality. This is what the F2 hybrids look like in terms of offspring output. Very few offspring and a cracked open fly host, whereas the controls, we see a lot of pupa that are alive in this case. There's about an 80 to 90% reduction, so in the larval state, we don't see any mortality, but as we get to the pupal state, they pass through this zone of death, essentially, and the larvae die at a significant rate. Okay, so the curious thing about this is that Nasonia has been studied from a genetic perspective, and this F2 hybrid lethality for about 20 years. People have been trying to find the genetic basis of this hybrid lethality. We have 5 chromosomes in Nasonia. They have been mapped. Those regions that associate with hybrid mortality are scattered across 4 chromosomes. But of course, this is just the genetic side of it. So what about the microbial side of it? Well, as we've seen, the hybrids have a different microbial community state than the parents. And in fact, we can say specifically that a Proteus genus is a rare microbe in the parents, but it proliferates in the hybrids. And what's interesting about Proteus is that it's a swarming microbial species, and swarmers often resist eukaryotic encapsulation and immune responses. So this is broken through the system from a rare microbe to now a dominant microbe. Okay, so can we test a causal association between this change in the microbial community and the hybrid lethality? And there's a pretty simple low-hanging fruit set of experiments to do here with 4 predictions. First, non-hybrid Nasonia will live. Conventionally reared hybrid Nasonia will die. Germ-free hybrid Nasonia, if we can cure the microbiome and they cause the lethality, will live. And then inoculated hybrid Nasonia, if we put the bacteria back into the system, they will die. So what do we see? Well, we need to develop a germ-free rearing system to do this, to remove all of the microbiome, and then put it back in to do these experiments. So this is the Transwell assay that my student Rob Brucker developed, where essentially we take eggs from Nasonia and put them on a media that's derived from the flies. It's hemolymph, it's sterilized, and now the eggs are taken out of their conventional environment and put into this Transwell system. In doing that, we can generate a significant amount of development from the egg stage to the pupa stage. And remember that the mortality occurs during the larval state. Okay, so the conventional hybrids died about a 90% rate. The germ-free hybrids, in this sort of Frankenstein moment, come back to life because we remove the microbiome, which would indicate that they're causal to the hybrid mortality. And then putting the bacteria into the hybrids in this germ-free state reinstates a significant amount of the hybrid lethality. Moreover, these QTLs that we talked about a minute ago are actually erased when we rescue the viability of these hybrids by germ-free rearing. Because now these hybrids live, and these QTLs are no longer associated with hybrid mortality. And the way we're measuring that is through something called a marker ratio distortion, which indicates alleles that are segregating unequally. We should see 50-50 ratios of Vitrepennis and Giralti alleles in hybrids, but when they're dying and those alleles cause the mortality, they aren't associated with the phenotype. So we have a switch from Mendelian ratios in the hybrid mortality states. Now, when we do the germ-free rearing and we rescue those hybrids, we restore those alleles to a normal Mendelian ratio. The control younger species pair, the Longicornis and Giralti, are compatible. There's no indication of hybrid lethality in this particular species pair, and there's no changes in the microbiome that are significant. So these live both in the conventional state and the germ-free state. All right, so we have these two cases in Nisonia. One is this microbe-microbe narrow sense incompatibility. The other is a broad sense microbe by genome interaction incompatibility. Wolbachia being this form of F1 cytoplasmic incompatibility, and the gut bacteria and host genome being this F2 form of hybrid lethality. I guess I want to end with some new data that's unpublished, and this is essentially a broader study of whether phylosymbiosis occurs more widely than Nisonia and other systems. And so we're doing that with Peromyscus deer mice, mosquitoes, Drosophila females, Nisonia females. And these are all gender-controlled, age-controlled, diet-controlled, etc. To sort of repeat what we did with Nisonia, but in a much broader vertebrate and invertebrate context. Yep, these little cute mice, but they're biting on their tails. They look a little uncomfortable here. And to make a further point, we're looking at whole adults. So we're looking at communities that are derived from whole adults of Nisonia, Drosophila, the larvae, and for the Peromyscus mice, we're doing fecal pellets, looking at microbial communities there. All right, there's been some work that have been done in these areas, and there's actually little evidence for phylosymbiosis. So the Schloss lab has looked at Peromyscus deer mice. There have been a couple labs that have looked at Drosophila, and there's no indication of phylosymbiosis, but these have also been done in uncontrolled states, either the natural population states or not well-controlled states in the laboratory. So we're going to give it another shot. And before I mention that, I should say that another great study on phylosymbiosis has been done by Thomas Bosch and Sebastian Frahn and colleagues, in which they were able to show that these several species of Hydra have co-varying microbial communities with respect to the evolutionary genetic relationships. Moreover, they could show that there was a distinct antibacterial peptide that regulated the microbiota relationships with their host phylogeny. So here's our data set. 5 different species complexes. The number up here indicates the number of species within each of those complexes, and each bar indicates a particular sample from many of those particular species. It's a large data set. There's not a lot you can take away from this slide, with the exception that hominids and Peromyscus tend to have more similar microbial communities as mammals relative to the invertebrates. And indeed, that's the case with these summary statistics for certain phyla of bacteria. Perhaps more interesting, though, is you can ask, how well does each sample's microbiota at a species level, an OTU level, a much finer level from the class relationship shown here, how well do those OTU level relationships predict the species it comes from, predicts the species it's derived from? And we've trained some microbiota models to show that you can highly predict where a microbiome is supposed to come from just based on computer modeling rather than obviously observing it ourselves. Some species do better than others, but these accuracy predictions of where microbiotas come from were pretty startling to us in this context. All right, you can also show microbial community structuring across host complexes. So these are highly dimensional data. We can reduce that highly dimensional data in a principal component analysis or in a network analysis. And you can see structuring in the microbial community across these 5 host hubs. Right? So blue is mosquitoes, purple Drosophila, green is the Nasonia insects, and these are structured individually and away from the red and yellow mammalian species. There's also structuring at a finer level within the host species. So not at just this broad clade level, but as you go within the species, you can see structuring. So now each one of these colors represents a distinct species within the host clade. And for some species, it's better than other, but in all cases, we see significant degrees of clustering of microbial communities within these particular host clades. So intraspecific microbiota variation is less than interspecific microbiota variation. There's another question we can ask with this kind of data, which is there a Goldilocks zone for detecting microbiota differences between animal species? In other words, maybe a species complex has had too little divergence in order for us to detect this pattern of microbiota variation being less within species than between species because those species are too young. It also could be that those species are too old, and so the microbial community relationships break down. So if we put the spectrum of divergence of these host clades onto this sort of Goldilocks scenario and then ask, where does the best data fit in? What's just right? This is what we see. And so the short answer here is that the level of divergence of the particular host species complex is correlated to the level of interspecific distinguishability of the microbiota. And this actually surprised us. We did not expect to see this kind of gradient. We thought it might just be some sort of sweet spot, but in fact, there's a nice correlation with time here. So the younger species of Nasonia, which diverged a couple 100,000 years ago to 1 million years ago, has significant but the least distinguishability in their microbiota, and then you can carry this forward all the way up to the mosquitoes, which have the highest divergence levels and the highest level of microbiota distinguishability. This is consistent with the phylosymbiosis model, being that there's an interaction between host genetics, divergence of those host genetics, and the microbial community assembly that occurs within those hosts. Okay, the central question here, though, is there a parallel phylogenetic or tree-like comparison here between the host phylogeny and the microbial communities? And we use two statistical tests to ask how similar are these tree-like comparisons. This is a phylogenetic tree. This is a dendrogram of the microbial communities. And to boil it a lot down, I don't want to get too lost in the details here, but in each species complex, we see significant degrees of phylosymbiosis. It's not always perfect, but we see significant degrees, even in cases where it hadn't been reported previously. So Peromyscus under these controlled rearing conditions gives us excellent phylosymbiosis with the exception of one particular species. Even Drosophila, 2 out of the 6 species we measured are off of fillet phylosymbiosis, but the rest are. And this is a relatively new piece of data with what we've seen so far in the literature. And across these time spans, whether you're a young species complex or an old species complex, we see significant degrees of phylosymbiosis. So there isn't necessarily an age dependency here for this level of analysis. We can repeat these analyses ad nauseum. We can use different OTU cutoffs if you know microbial community analyses. We can use different ecological metrics. And the same results tend to spit out over and over and over. I would say that Drosophila show the most tentative evidence or the most variable evidence for phylosymbiosis, and the remaining 4 taxa show strong evidence. Okay, so much like in Nasonia, we can ask, is this pattern consequential or not to a vertebrate system like these deer mice? And in this case, postdoc Kevin Cole is now moving fecal material and the microbial communities associated with the fecal material into one common recipient. And essentially what he was able to show is that mice from this species receiving microbiota from more distantly related hosts down here exhibit decreased digestive performance. And that's just a measure of how much food is eaten and how much poop comes out. And so digestibility is how much poop or how much food is being stored and not pooped out. And so this is an indication of if you digest more of your food and you use it, then you have increased digestive performance. And so this is a finer scale analysis. We've also seen in mice that if you move a human microbiota into a mouse gut, there can be severe compromises in the immune system and they're susceptible to enteric infections. But even among these closely related species now, Peromyscus, we can see problems with digestive performance. And what's interesting is also Kevin measured the response. How do the mice now react when they have a different microbiota and they're not digesting as well? And one way you react as a mouse is you start eating more. So you measure food intake, grams of dry food presented minus grams of dry food remaining overnight. And so the resident species with its resident microbiome has a lower food intake because it's digesting more of its food relative to those that received a foreign microbiota. So there's a behavioral response now to receiving these gut microbiotas that associate with decreased digestive performance. Okay, let me sum up. So based on this unpublished work, there's somewhat of a host-specific uniqueness to an animal microbiota assemblage, right? That these are somewhat personalized with respect to species. That is, clearly a mouse and a Nasonia have different microbes, but even within Nasonia, the multiple species within Nasonia have different microbial communities. And they matter both from a relationship perspective as well as functionally, because we can reduce survival with different microbiota transplants, and we can reduce digestive performance in the mice. So these are not just relationships, but they are functional relationships allied with host evolutionary history. So does this have an applied relevance? I want to come back to this slide because it indicates that there is a relationship between host genetics and microbiome similarity in humans. And so one might sort of speculate at this point that these microbiome differences and genetic differences might be uniquely tailored. That is, your microbiome may be a lot better for you than your neighbor's, for example. And whether this needs to be taken into account in terms of a personalized medicine, a personalized microbiome as well as genome seems relevant. Okay, so summary of the findings. We have this Wolbachia case in Nasonia. Other studies are coming on board from flies in particular. We have John Janicki's work in the mushroom-feeding flies. We also have Wolfgang Miller's work, which is focused on the old study by Lee Ehrman that I initially mentioned. And they were able to identify the bacteria that cause the hybrid sterility are in fact Wolbachia bacteria. We also have this gut bacteria work, which is extending into other systems where phylosymbiosis appears more common than just in Nasonia or Hydra, for example. Okay, I want to thank everybody for listening. I want to thank the key members of this team. So Rob Brucker did a lot of the Nasonia work, hybrid lethality work in developing the germ-free system. Kevin Cole is a vertebrate microbiome expert. He did all the Parameciscus work. And Andrew Brooks and Teddy Van Opsel are now working on some aspects of phylosymbiosis as well. Funding from Vanderbilt, the NSF, and the NIH. I think I've got left a few minutes for questions. Thank you very much.
Yes, thank you very much. That was highly interesting. I'm pretty sure there are questions in the audience. Then.
How about the Nasonia? Is there an interaction between Wolbachia and the gut bacteria?
Yes, this is a great question. And yeah, so the question is how about in Nasonia, is there an interaction between Wolbachia and the gut microbiota? In other words, does Wolbachia shape the gut microbiota? And the short answer is, I don't know, but I love the question. We've thought about it. The longer answer is, Wolbachia do cause immune responses in insects. We know that they prevent viruses from replicating, such as the dengue virus and Zika virus. So it seemed quite natural that they affect their bacterial communities of the host as well. I think that one aspect that I'm particularly interested in, it's a little bit of an extension of what you just asked, which is if you remove the microbial community and leave Wolbachia behind, and so you only have a Nasonia and Wolbachia, what does that thing act like? And does it cause, for example, cytoplasmic incompatibility? Does it do the things we normally think it does as a way to functionally look at, is Wolbachia dependent upon the microbiota, or does it shape the microbiota, and vice versa? Thanks for the question.
So when you say germ-free, is it free of both Wolbachia and the gut bacteria?
Yes, it is free. So the question is it free of Wolbachia and the gut bacteria in a germ-free state? And it is, yeah. Because the Wolbachia cause a form of cytoplasmic incompatibility between the species. So if we had Wolbachia there, we couldn't ever interbreed them to get the F2 hybrids. So they have to be gone as well, yeah.
Thanks. Who's next? Question.
Well, I think I can do like if you change the gut microbiota very quickly in your experiments, right? So you just do an immediate change. What if you would change them more gradually over longer time periods? Could you understand? You have to repeat it.
I do. The question, I want to elaborate a little bit, but the question is what if you change the microbiota a little bit later on in time or development?
Just over a longer period of time rather than just doing it quickly in a single spot of like time spot. If you do it over over development or over weeks or whatever.
Yeah.
More like if you go into a holiday, you might change your gut microbiome too, right? If you go to India, you're probably going to have a different one afterwards.
Yeah. There's a lot of answers to that question because dynamically the microbiota is changing over time. And we haven't followed it through the aging process, which at least in humans, we know that the elderly have a different microbiota than adults. So we'd expect that to happen. The other way to sort of dynamically think about that is what happens over multiple generations. So when we put a foreign microbiota into Nasonia, what happens to the next generation? Does it sort of retool it? Does it come back to its native state? And these are questions that we would love to know the answers to. But there's a lot of ways to take that. The most important thing, I think, from my perspective is what is the role of transmission of the microbiota from one generation to the next? And perhaps aging and time affects that, because if older females reproduce, do they reproduce a different microbiota than, let's say, younger females? But fundamental to that is what's the fidelity of these microbial communities across generations, and how are these animals passing them on? If it's not vertically in the egg, like Wolbachia, is it perhaps on the surface of the egg? Is it perhaps a fecal pellet that's left behind for these offspring to feed on, and so forth?
Thanks.
Thank you for this interesting talk. And my question is about the correlation between the microbiota similarity and the host similarity. I was wondering if you can focus on specific genes, gene networks, functional networks in the host to increase the correlation, basically.
Yes, great question. So we are working on that. Basically, we would like to find what are the host genes that are regulating phylosymbiosis, much like the beautiful Hydra work. Our approach to that is going to be sort of in a reverse approach, which is we want to know what are the genes that are causing the hybrid lethality that interact with the microbiota in the F2 hybrids. And once we identify those genes, then we're going to work backwards into phylosymbiosis and ask, are those the same genes that regulate the phylosymbiotic pattern? We do have some gene expression data that suggest the genes involved in the hybrid lethality are in the prophenyl oxidase pathway. So either serine proteases, which splice prophenyl oxidase, or prophenyl oxidase itself, because those genes are misregulated in the hybrids. What's interesting is that the melanization response that occurs in those hybrids, they darken up. The melanin is produced through the prophenyl oxidase pathway. So it looks like a pretty good candidate for tackling with RNAi knockdown technology to figure out if those genes are there. I just need somebody to work on it. That's what we're waiting for. Yeah.
Right, Seth, thank you for your presentation. So your concept of phylosymbiosis states that host phylogeny varies with host microbiota, right? And this is based on diversity studies, probably using the 16S rRNA gene as a marker. So from a microbiology perspective, the next relevant question would be, how does that relate to the functional gene repertoire of this holobiont? And how would that translate into functional questions?
Beyond what we've shown, because we've done, obviously, these transplants to look at the functional work. So what else would you want to see there?
The microbial gene repertoire, the microbial metagenome, because showing change in diversity may or may not mean a change in the functional gene repertoire. So does it have consequences?
I see. Yeah. So we have predicted metagenomes from these communities. We don't have the actual metagenomes. The predicted metagenomes show very little differences between the microbial communities. I don't take a lot of weight in that data because the predicted metagenomes are based on very conserved, GO categories or COG categories that only highlight the most conserved differences. So far, I think that that's not going to be a useful way to go about it. The variation there would be illuminated through metagenomics and sequencing these highly variable regions. of the microbial community. We are also now pulling out individual microbes from each species and sequencing their genomes to look at fine-scale variation between, let's say, a Proteus in Geralti and a Proteus in Vitropenis to see what the fine-scale differences are. I think probably beyond metagenomic prediction, the most relevant aspect is phenotypic function. And so we can get a lot of answers just with transplanting communities rather than inferring what might be the differences. Yeah, okay.
Anyone else? Am I allowed as a snail person to ask, did anyone look into mollusks? You talked about arthropods. Mollusks are the second largest phyla after the arthropods. Did anyone look at those animals?
Which one?
Mollusks?
Mollusks. Not that I'm aware of, but I'm not a mollusk expert, so maybe somebody else can help.
I'm disappointed.
I think, Matthias, that's what you can do.
Yes.
Yeah.
All right. You're very on time, so
It's time to drink.
yep.
All right. Thanks.
Thank you very much again.